He started.
They're a committee.
No,
Right, so, yes, you have to stand. Right, so everyone, sorry about the technical issue, we forgot to record and some of the committee members denied on this recording, so we're gonna redo again, sorry about that.
You got some practice, it's okay.
Yeah, it's fine. You warmed up.
You don't even have to be there for it.
They don't have to, but I
think it's
like,
If I go back to the first slide,
yeah.
sorry about that, I should have noticed.
Oh, no, I should have noticed too. I'm so sorry, guys. That's my bad.
And you can speak a little bit louder.
Speak a little bit
Yeah.
louder. My bad, y'all. Happy Friday.
You told me some things already, so I'll just reiterate it.
Yeah. That's right, okay. Okay. So. We're,
Still good?
yeah,
Right,
okay,
no introduction.
all right, yeah, sorry about that, guys. So we forgot to record, so that's my bad. So I'll start from the top. Okay, so I'm John Ertle. I'm excited to talk to you about my PhD research, managing tipburn in controlled environments, specifically within indoor farms for lettuce production. So just to get right into it, controlled environment agriculture is a method of growing food crops under protection. And so we modify the environment to prevent some of the abiotic stresses that you might encounter in the field setting, and also increase the growth rate and quality of produce grown under some kind of modified environmental structure. A range of CEA can be anything as simple as a skeletal structure on the left side with a film wrapped around, still allowing in sunlight, but protecting from some of the abiotic stresses, all the way up to totally enclosed greenhouses with under glass, and then as well as fully enclosed indoor vertical farms, usually inside of some kind of building or modified building structure. So indoor farms have been rapidly growing in the US as a sector of the industry over the last 15 or so years. For many key beneficial reasons, including that we can localize production with this type of strategy, placing these farms close to food deserts, urban centers, other areas from which we want to market to those local consumers. We can produce fresh, high quality food due to the environmental controls, and then provide fewer chemical inputs to prevent things like pests and seeds from occurring in cropping systems. It also allows us to reduce the number of food miles used in transportation of this produce, since we're able to produce them highly locally to various consumer environments. It also allows us to supply local jobs to those regions. And due to the structure of these farms, we often are able to stack these growing systems vertically on top of one another, which can allow us to have up to 100x more productive farm than comparative open field area. So the square area of the indoor farm can be 100 times more productive than the open field due to the vertically integrated structure, and also the ability to produce crops year round without any breaks. Many of these growers also use hydroponic systems, which is the replacement of soil systems with a weak fertilizer nutrient solution that's constantly circulated and roots are submerged in that system. And that, because we're able to use this as a growing media, we can use less than 90% of the water, or I'm sorry, use greater than 90% less water than open field applications. Although this type of environment, being highly controlled and highly productive, allows us to have a really robust production of many food crops, most of these growers produce lettuce in this type of environment. However, we often run into a problem of lettuce tipburn, and that we have this healthy looking lettuce plant on the left, but on the right, once you start peeling back the outer leaves, you may see marginal leaf necrosis happening towards the center of that compact head. And this greatly reduces the marketable quality and can also limit the yield of the crop, as we are limiting leaf expansion and biomass accumulation due to this deficiency. So lettuce tipburn itself is generally caused by a breakdown in the formation of pectin. So pectin is a critical structural component of the cell wall. And when we start to have deficient pectin formation, the cell wall integrity is at higher risk of being broken and the cell lysing. So this is generally caused by the lack of this calcium cation as a linkage structure between these polysaccharide chains. So pectin itself is just this polysaccharide chain linked together to give functional strength to the cell wall. But when we have a deficiency of calcium, this pectin starts to break down and we start to have the cell wall collapsing. So calcium uptake is rather important to understand in this application. So calcium and water are transported together from the roots to the shoot biomass of the plant through the xylem. And so it's taken up here in the root system and then pushed upwards into the above ground biomass. And this uptake is called mass flow and driven by primarily transpiration during the daytime. So the size of the water droplets is somewhat relative to the amount of transpiration happening from different leaf tissues of this plant. And so when we have a smaller amount of transpiration occurring in any one area, we may have less accumulation of calcium. During the nighttime, this mass flow is driven primarily by root pressure. And so the leaves of the shoot will close stomates and then prevent transpiration. And so that we have an increase in root pressure driving that mass flow upwards. Calcium is also an immovable nutrient, meaning that once it is requisitioned and used in one area of the plant, it cannot be taken away and translocated to another area of high demand. And so we typically will run into this deficiency in the young leaves, which are getting much less calcium than some of the other tissues. And then there's no way for the plant to essentially rescue that tissue danger of the calcium deficiency. So it's important to understand the growth rate of lettuce in the context of when you would likely see a calcium deficiency. So on the y-axis is the weight shoot biomass of the lettuce head during a typical 4-week indoor farm production cycle. And then the different colored lines representing different light intensities between 9 and 19 moles per meter squared per day, which is the summation of the daily light received by that crop canopy or the daily light integral. So when we increase that daily light integral from 9 to 19 moles per day, then we see an increase in shoot fresh biomass of 52%. And so the shape also from week to week is important as this growth rate of lettuce increases exponentially over time. So the relationship of calcium with leaf tissue is essentially a supply and demand function. So the supply of calcium is driven by that root pressure during the nighttime and transpiration during the day. And the calcium demand is primarily related to the growth rate of lettuce. So as that relative growth rate increases over time, like I mentioned, we start to see this deficiency occur somewhere between 20 and 28 days after transplant into a farm setting. And this is really similarly a function of calcium demand. So when that demand for calcium becomes too high, we start to see the deficiency occur in these young leaves, which are lower transpiring than these other areas of the plant, such as outer leaves. So to understand a little bit about how transpiration functions in the plant, transpiration is basically evaporation of water from the leaf tissue, and then the rate is regulated by the stomatal opening as these stomatal pores lining and dotting the leaf surface will open and close related to the perception of the plant of water stress and also the aerial conditions, but it can be driven by a number of environmental factors, what that rate of transpiration looks like. And so some of those factors would include the airspeed or the wind over the crop. Basically, as more air is being moved throughout the crop, the more transpiration is going to happen from that leaf surface, as well as the humidity. So when we have a higher aerial humidity, we're going to have less transpiration as there's a very high humidity condition within the leaf. And as water moves out, it's going to move out much faster when there's a low humidity condition outside the leaf. We also have the input of the air and leaf temperature. And so basically when the temperature is higher, generally there's going to be more transpiration away from the leaf, as that loss of water through transpiration is going to leave some of that heat energy. And then the input of radiation, so more radiation is generally going to equal more transpiration in the plant. However, the light quality matters in this context, as we have sunlight, which is going to drive much more transpiration from the leaf than something like electrical lighting, because the sunlight wavelengths expand far into this near infrared region, far beyond the range of photosynthetically active radiation, which is very highly absorbed by these leaves to be used in photosynthesis between 400 and 700 nanometers. And it's also typically the range of light quality that we put into the vertical farm setting from something like LED light fixtures. But sunlight drives somewhere, because 50% or so of that sunlight is near infrared region, we're going to drive much more transpiration from leaves when they're from under sunlight versus under electrical lighting. We would measure this photosynthetically active radiation in micromoles per meter squared per second, which is the photosynthetic photon flux density. So in 3 different lettuce cultivation environments ranging from the open field to the indoor farm, we're going to see a higher contribution of a lot of the environmental conditions to drive transpiration in these different settings. And essentially from left to right, we're going to see a lower relative transpirational flux occurring for these plants due to the change in how those environmental conditions are being generated around the plant. However, that's essentially the inverse of the relationship with relative tip burn risk. So as we have a lower transpiration occurring, we're going to have a higher tip burn risk in these different settings. So there are several ways to prevent tip burn, but once it occurs on the plant, it cannot be rescued. So when we have tip burn occurring on these leaves, we're limiting that leaf expansion into biomass, but there's no way to reverse that necrotic tissue back into the healthy functional tissue. And once that deficiency generally occurs, we usually see each newly initiated leaf afterwards also developing the same calcium deficiency. So some common strategies to prevent this can be cultivar selection, selecting low tip burn risk cultivars, enhancing the transpiration via mass flow, harvesting the crop early to avoid the deficiency entirely before it usually occurs, also slowing down the plant growth to limit the calcium demand, spraying an application of foliar calcium so that there's an exogenous supply of calcium available at the leaf surface, and then also utilizing high nighttime humidity, usually greater than 95% relative humidity during the full nighttime to drive root pressure and mass flow of calcium. However, there's some drawbacks to these various methodologies, especially for indoor farms. So while selecting low tip burn risk cultivars is an excellent way to avoid tip burn in your indoor farm, it's still been a major topic of interest to understand and dissect some of the tip burn resistance genetics and seeing what the germplasm can offer in terms of finding a better way to finding resistance to tip burn. However, currently, commercially, the 3 of the larger seed companies for lettuce marketing to indoor farms have double or triple digit offerings in the number of lettuce cultivars. And this can be a range of different morphological types, leaf color, leaf shape, texture, nutritional quality, appearance, many factors. And so one of the biggest hiccups here is that we don't fully understand tip burn resistance yet. And then once we do have a really good foundational understanding of what genes are important in this relationship, it's going to be challenging to address this tip burn resistance to this many currently offered cultivars. So one of the main value adds of the indoor farms is that with that short production cycle, we can rapidly change the types of lettuce grown in the environment to meet consumer demands or market demands consumer target audience. Additional strategies to prevent tip burn. Like I said, we can harvest early under typical 4-week type planting with 40 plants per square meter, we can match the same amount of biomass produced in that 4-week cycle by having 2-week plantings at 272 plants per square meter in the same indoor farm condition. We can reduce the growth rate by reducing the light intensity or reducing the temperature in that growing environment. You can also apply that exogenous foliar calcium. However, in these indoor farms, harvesting early will increase your material and labor costs to reach the same biomass target. Reducing the growth rate can reduce your single crop revenues. You can produce less biomass, and then also your annual revenues. You may have less harvest per year, or you might have total loss in biomass production over that course of the year. And then also foliar calcium sprays have a few issues. Is one, most growers don't want to wet plants in the growing environment, as well as the potential to be spraying this application, damaging expensive electrical equipment, damage to structures within the farm, and then also potentially reducing yield of the lettuce due to a reduced gas exchange until that water has been cleared from the surface. We can also enhance the transpiration in the farm, but this comes with some difficulties as well. So reducing aerial humidity during the daytime is one effective option. However, we also rely on the HVAC systems equipped to these farms. And as these plants are constantly transpiring into that environment, it becomes expensive and challenging to manage this humidity to reduce it enough that we're going to be driving much more mass flow of calcium to those leaves. And then additionally, because of the vertically stacked structure of these farms, we have competition within that aerial region with lights and other sensors and equipment that it's really challenging to increase airflow over that crop canopy. Specifically, we've known that downward vertical airflow that breaks into this inner compact head structure is really, really effective at relieving tipper risk. But we also have to come up with unique designs that can fit within the various custom built structures that often are used to create these different growing areas. And so while something like a perforated air tube design has become something that's of high interest for growers to have developed, often the unique style in which they built these growing rack structures provides a limitation in that it's really challenging to get that adequate amount of airflow in that direction. Also, that elevated root pressure technique of elevating that humidity at night to drive that root pressure of the plant has been also shown to be effective and can reduce the tipper quite considerably when that dark period humidity is above 95%. However, we run into the same issue where we're humidifying and dehumidifying a farm every night. That can be a real expensive cost if you're managing that environment with HVAC or other electrical equipment, and then also most equipment is not outfitted for that type of application. And then also you might have things like condensation on various types of electrical components within the farm. That can be also a challenge if you're going to jeopardize the very expensive equipment you've purchased and installed within this growing environment. So we wanted to run a series of experiments to better understand some of the tipper dynamics in indoor farms. So the first experiment we ran was to standardize some of this tipper testing information. So we thought we could develop a tipper inducing condition to evaluate this risk of growing lettuce in indoor farm conditions. Seeing as there's so many cultivars available, we wanted to get a better picture of what that would look like for a normal grower. We thought that the more enclosed head type varieties like butterhead varieties that also have a compact head structure, like I showed in the earlier slides, were romaine varieties with the very tall outer leaves that also can be enclosing upon the center of that lettuce, would have a relatively higher tipper risk than those with a more open head variety, like many of the leafy types, where you can actually see the meristem throughout the entire growing period. Also, one of the main reasons we wanted to work on this tip burn testing standardization is that most of these seed companies developing lettuce cultivars are evaluating their tip burn risk under greenhouse conditions, which have a lower risk of developing lettuce tip burn than indoor farm conditions do. So we wanted to have a more standardized direct approach to evaluating this risk for indoor growers. We also explored 2 different experiments trying to mitigate tip burn under the initial tip burn inducing condition we wanted to develop. So in the second experiment I'll go into later, we reduced light intensity over the crop canopy to reduce that tip burn severity, but we knew we would also reduce some of that biomass introduced. But even though we might lose biomass, we think the strategy could be profitable if the reduction in tip burn is greater than the amount of biomass lost. And that this strategy was probably going to be most beneficial for higher high tip burn risk, as it might be better to produce a smaller head with little tip burn than a larger head with a very severe tip burn. Then the third experiment that we explored was a unique application of nighttime lighting to try and prevent tip burn. And this was really trying to drive a little bit of transpiration during the nighttime to enhance mass flow of calcium without contributing to the additional growth of the plants. As calcium demand is basically zero during the nighttime, this would be an interesting period to try and explore, similar to how that root pressure technique is only used during the nighttime. We want to drive lettuce mass flow when there's no demand for calcium. We thought that nighttime light quality used during that period would be important in terms of how much transpiration we're able to drive. So we thought including red light in that spectrum is going to be more effective than driving some other wavelengths within the typical R range. And also we thought that nighttime lighting will likely be able to reduce this tip burn severity by driving that mass flow of calcium. So the experiment itself was basically the same throughout the different experiments, but essentially how this worked, we would seed on day 0 and grow the seedlings for 2 weeks. On day 14, we would transplant plants into a hydroponic system. They were a bit smaller than this at the time of transplant, but then we would grow them for 4 weeks under this crop production cycle until harvesting on day 42. So 28 days after transplant. So rather typical indoor farm type of production style, and then growing in those NFT channels with nutrient solution constantly being circulated throughout. So I see the growth took place in growth chambers for those 2-week period, and then the crop production was conducted in 2 separate walk-in growth chambers that were set up identically so that we could use different experimental designs within the 2 and across the 2 chambers. We had 4 carts per chamber with NFT systems set up and 28 plants planted per, or 24 plants planted per cart. It's important for us to understand how we're affecting the rate of tip burn. We have to measure the rate of tip burn. Seed companies often report tip burn incidence, much like a pathology measurement. Say things like this lettuce is very strong against tip burn, very resistant to tip burn. But often these seed companies are not sharing the metrics at which they were able to diagnose, develop their tip burn rating. And so for most growers, it's basically impossible to compare the relative tip burn risk from one company to another. In literature, there's many different ways to evaluate and assess tip burn, but that are not consistent across many of the literature reports. But the 3 main ways in which you can evaluate tip burn of lettuce is a measure of incidence for the whole crop, so a percentage of how many plants are affected by the deficiency. Also the severity, this is on an individual plant basis, so like a percentage of leaves, percentage of leaf area, or a visual index that was developed for ranking tip burn severity. And then emergence, which is the amount of time that's elapsed before the tip burn symptoms appear on an individual plant. So we took many measurements throughout the experiment. We did fresh and dry mass of the roots and shoots at the time of harvest, as well as leaf count for tip burned and non-tip burned leaves. And then At harvest, we also assessed with a mineral composition analysis of the shoot dry mass to evaluate differences in leaf mineral concentrations between inner and outer leaves. Inner leaves being somewhat relative as to how many leaves was considered inner based on the different cultivars and how many leaves they produced. And then we've also measured gas exchange throughout each of these experiments with the Cyrus-3 developed by PP systems. And then we measured and reported different incidence, emergence, and severity throughout the experiments. So to standardize this tip burn testing, we wanted to grow the commercial cultivars under that tip burn inducing condition and identify the relative risk. This risk is going, for instance, we're inducing the tip burn in this condition, it's going to be all the lettuce will have a higher tip burn severity than you would normally see in an indoor farm setting. Also want to identify the differences in severity between lettuce of different morphologies, so different types and colors of leaf color of lettuce. And we wanted to evaluate the severity against the recommended production systems proposed by the companies that developed them. So our tip burn inducing conditions are here on the left side, but we had a 16-hour daytime and an 8-hour nighttime. During the day and night, we had a 23 and 19 degrees C set point, as well as a relatively high relative humidity during the daytime, about 75%. And then we vented during the nighttime to less than 60%, which translates to a vapor pressure deficit of 0.7 during the day and 0.88 kilopascals or less during the night, which should be or greater. The CO2 concentration in the daytime, we injected CO2 to enhance the growth rate at a rate of 1,000 micromoles per mole or 1,000 ppm. And then the photosynthetic photon flux density was 290 micromoles per meter squared during the daytime, and then no light during the nighttime, translating to a daily light interval of 16.7 moles per meter squared per day. And we also had a very low horizontal and vertical airflow rate of less than or about 0.07 meters per second in both of those directions. So this relatively high humidity condition, this relatively low airflow rate, as well as a high light intensity and high CO2 concentration, we're trying to drive a fast growth rate, but also reduce the amount of transpiration occurring in the farm. We used LED grow lights within this chamber that were adjustable, and we targeted that PPFD of 290 with this light quality spectrum, so about a 2 to 1 to 7 blue to green and red ratio. And this what lights look like. This rather purple style is rather typical for a lot of indoor farms, mostly due to the efficiency of red and blue LEDs being preferable to use for energy consumption purposes. So we selected 10 cultivars grown commercially. They were a range of butterhead, romaine, and leafy type varieties with both green and red leaf colors. And then they were recommended by the seed companies for various production systems, either indoor, meaning greenhouse and indoor farms together recommendation, outdoor, traditional open field cultivation, or both indoor and outdoor settings. And to jump right into the results of what this looked like, we did see similar tip burn emergence across the 10 cultivars here on the x-axis. This is the number of days after transplant or time to tip burn. And we see that the average here was somewhere about 22 plus or minus 2.6 days before we started to see tip burn symptoms in each of these 10 cultivars. There's only one cultivar that had a much later tip burn emergence, but for the most part, all of these cultivars got tip burn around a similar period of time. Tip burn severity was a bit more variable. So in this case, it's percent of leaves on the y-axis affected with tip burn is calcium deficiency, and that average was about 40 plus or minus 18% of leaves. There were a few cultivars that performed rather well and had low tip burn severity, but for the most part, we saw varying degrees of tip burn from cultivar to cultivar. If we aggregate these together and look at them as their categorizations into butterhead, leafy, and romaine lettuce types, that tip burn severity actually didn't vary much across those three types at all. So while we thought initially and colloquially it's relatively understood that the more enclosed type head varieties are going to have a higher tipburn risk, but in this selection of cultivars, we didn't see any differences. We also didn't see any differences between red and green type lettuce. But we did see lower tipburn severity in cultivars recommended for indoor production. So that's good news to us, and that there's some selection pressure happening at the level of the seed company to both recommend and I think hopefully develop some lettuce that's more resistant to tipburn in this indoor vertical farm setting. We did also run a follow-up trial where we kept 3 of those initial cultivars and grew them again under a tipburn-inducing condition. And then also 17 new commercial cultivars that were either grown by stakeholders as part of our grant, or were recommended by seed company for indoor production, or recommended by a third-party company, the seed seller for indoor growers as well. And what we found here were there were 5 out of 20 of these cultivars had less than 10% tipburn severity, which indicates to us that there is a good number of cultivars that exist in this selection that are relatively low risk for these indoor growing environments. So once we went through this trial, we wanted to select 2 cultivars to retain and use in the future experiments. So we picked Clee, which is a leafy, more open structure red type variety, which was the most severe tipburn, highest tipburn severity in that initial trial, and then also Rex, which is a moderately sensitive green butterhead variety. So then we focused on tipburn mitigation methods. So in this experiment, we were looking at the end of production lighting application to prevent tipburn. And so we wanted to reduce that DLI during that fast growth phase to see if we could lessen the amount of the calcium demand during this period where we typically see a deficiency occur. We also wanted to quantify that the reduction in tipburn we might see, and then also that that yield of those plants, that we could see if there was a potential point at which that reduction in yield and reduction in tipburn severity would be preferable to provide an economic return of this reduced lighting strategy. So, like I brought up before, the growth rate over time is essentially a function of increasing calcium demand. And so we wanted to decrease that calcium demand by reducing the light intensity during just this period of higher tipburn risk. So knowing that this typically happens around 20 plus days after transplant, we decided to initiate these lighting treatments at 16 days after transplant to preempt the onset of this deficiency. So we grew Clee and Rex under 4 different lighting conditions, where the conditions leading up to day 16 were exactly the same: 100% light intensity, which is about 17.4 moles per meter squared per day. And then for from that point onward, for the last 12 days of growth, we reduced light intensity by to be either 85, 70, or 55% of that original 100% intensity. What we found here was that as DLI is reduced on the y-axis, the shoot biomass decreases on the, or I'm sorry, x-axis. The shoot biomass does decrease with that reduced DLI lighting, up to 22% in Clee and 26% biomass loss in Rex. This was also able to affect our tipburn severity, but it was a bit cultivar specific in how this played out. For Clee, until we got all the way down to 70% of that light intensity, we had similar rates of tipburn severity, but then it drops off considerably down to at 55% of that light intensity. For Rex, it was a more linear response, and at the lowest lighting treatment, we actually eliminated tipburn entirely for Rex. So it seems like it's a possible strategy to reduce that tipburn in this setting. However, even though we reduced that growth to 22 or 26% and tipburn by 75 or 100%, we still needed to determine if this was going to be a profitable strategy. So on the y-axis here is the contribution to the profit margin for the farm. 100% is our baseline, so there's going to be no information there. But for Clee and Rex both, as we reduce that light intensity, we do increase the electrical savings as we have reduced power consumption of those electrical lights. However, the biomass production was so large for these cultivars that as we reduce the light intensity, we're reducing the yield so much that the profitable, or that the revenue generated by these plants is decreasing. Even though we're taking out the amount of, even though we're able to salvage a lot of that biomass due to lower timber severity. However, for CLE, the highly sensitive cultivar under 55% lighting, we are able to come up with a increase in the profitable revenue of crops grown under that lighting strategy by about $2.55 per meter squared. So even though this strategy doesn't seem profitable for most cases, it is possible to have an increased economic revenue under reduced light intensity for CLE. So this strategy could be profitable for highly sensitive cultivars and application. Right, so I'm going to talk about experiment 3 as well. So this is the unique application of dim lighting during the nighttime. So we wanted to apply this light during the night and see what kind of gas exchange responses we would get from the plant if we're able to drive that transpiration. We wanted to increase that max flow of calcium during that period of no calcium demand and see if the strategy could mitigate that tip burn risk during the nighttime. So we grew them again under the same tip burn inducing condition we initially developed, and then added in this dim lighting component for the 8-hour nighttime. But we're trying not to contribute to the calcium demand. So it's important to understand how much light we're able to add to the plants before we start increasing that calcium demand. So on the y-axis is the photosynthetic rate, and then the x-axis is our photosynthetic photon flux density. And then this is a simple generic light response curve, right? So during the nighttime, when there's no light, there's going to be dark respiration occurring. The plant's going to have a negative photosynthetic rate. As we add light, the plant will increase in photosynthesis. At some point, we're going to reach the light compensation point where the rate of respiration and the rate of photosynthesis is the same. And above that point, we're going to be driving active growth of the plant. So we're going to increase calcium demand over time or under higher light intensities. And then at some point we'll reach the light saturation point where the maximum rate of photosynthesis is achieved. But this indicates to us that we need to find this light compensation point and then apply light low enough to not drive above that compensation point. Also important to understand what light quality we want to work with in this context. And we wanted to explore a couple different light qualities here that have been reported in the literature, but not really explored in many applied application settings. So I won't make you read this whole physiological mechanism, but basically blue light has been reported to activate the phototropin in guard cells, driving a phototropin mediated response that causes the guard cells surrounding the stomates to swell and open that stomal aperture to allow transpiration to happen. And this blue light effect has also been reported to be enhanced by the effect of red light specifically on the guard cells. So as red light is also entering these guard cells, it starts driving photosynthesis to reduce substomatal carbon dioxide. And both of these mechanisms, so the phototropin and photosynthesis mediated responses, are enhanced by one another. And so we thought that it would be interesting to apply this type of these two different types of light qualities to these plants to see if we could generate a response that was helpful in this setting. So the first light quality is 100% blue light. I'll just call B100, PPFD on the y-axis wavelength here. And then the second light quality was BR20, which is 80% blue and 20% red light. And so for CLE and Rex both, we've looked at light response curves. So this is just the very beginning of that curve. And the photosynthetic rate under B100 and BR20. And with either light quality for both cultivars, we found the light compensation point for these plants was somewhere between 78 and 155 micromoles per meter squared per second, depending on the light quality that was given to those plants. So that indicates to us that we as long as we stay below these values, we're going to not drive additional calcium demand. So we selected just we saw some responses as low as 30 micromoles per meter squared per second. So we decided to select these to drive to add this light at just that low 30 micromole intensity during the whole nighttime. Just to give you an idea of what that looked like from the daytime to nighttime transition, as we're reducing that intensity and changing the light quality and overexposing that camera. And then we wanted to be able to conduct multiple of these tests in different chambers. And so we had to cover all the walls with black lining and then also a curtain down the center so that we could get full exclusion of photons from one half of the chamber to the other, so we weren't influencing these treatments back and forth. So just go into the results, because we were able to add in such a small amount of light, less than 1 mole per meter squared during the entire nighttime, we didn't see any changes in yield between any of these treatments. And then we also didn't see any changes in the daytime gas exchange, as these plants normally functioned under the tip burn inducing daytime condition that we maintained without any changes. But we did see some differences in that nighttime gas exchange. So on the y-axis here is this amount of conductance, and then the 3 treatments, B100, B80, R20, and then the control, which is just an entire nighttime darkness. And we saw that for Clean and Rex, the responses were slightly different. Rex had statistically significant responses, and then Clee had a wider range, was not statistically significant, but the trend appears to be in the same direction. And so with B100 lighting, we were able to increase this amount of conductance about 50% or 45% for between the control and that B100 lighting with Rex. However, with either cultivar, once when we look at the transpiration rate under this BQF condition, we do see an increase in transpiration from the control to B100 with that nighttime lighting, even at that low intensity. But this did not sufficiently drive mass flow of calcium with these plants. So we didn't see any differences in tip burn severity between any of the treatments. And so they're similar amounts of tip burn regardless of the addition of this nighttime lighting. So we thought perhaps that the indoor condition that we had created with this really low transpiration rate and low airspeed was maybe inhibiting the mass flow of calcium, even though we were increasing the small conductance. And so we ran a follow-up experiment to look at the inclusion of B100 or nighttime darkness with the addition of downward vertical airflow fans that were going to blow air over the top of the canopy at a rate of 0.4 meters per second. And so when we did this, we found some interesting differences as well in this nighttime gas exchange. For Rex, although we had much stronger signal between B100 and control previously, we didn't see any differences in this follow-up experiment. And the fanned treatments are just denoted with _F and dark F being the control plus fans. For Clee, however, we did see a reduction in small conductance under these fan treatments, much more so than we did under the B100 or control treatment. So we don't see any real change between B100 and control at this level either for Clee. However, with the inclusion of fans, we were essentially able to eliminate tip burn entirely for any treatment in which we had fans, regardless of the nighttime lighting presence or absence. And there was just one plant with one leaf that had tip burn in this trial. So generally, this was a highly effective solution to be able to eliminate tip burn entirely, although the nighttime dim lighting component was really not as effective as we were hoping that mechanism might be. Just to give an example, if you can tell on this screen, but the treatments with no fans on the left, you can see tip burn just about the center of every one of these heads. However, on the right, we didn't have any tip burn occurrence among these plants. So to draw some conclusions from these various studies, with the tip burn testing, we were able to develop and test a standardized tip burn inducing condition for testing of tip burn severity in multiple lettuce cultivars. We found that the tip burn risk of lettuce is more relative to the cultivar than it is to its categorization of morphology or leaf color. And that the seed companies are improving and recommending cultivars that are having a lower tip burn risk under this condition, which is excellent to see that that development for better selection for indoor growers. With the end of production lighting, we found that reducing the DLI at the end of that production cycle can reduce our tip burn severity, but there's a associated yield penalty that's rather linear and predictable. But this strategy can create a marketable, a better marketable value than under full lighting production on a per meter square basis if we're using it and applying it to highly tip burn sensitive lettuce cultivars. So this makes it an application that's readily something that can further be tested and applied in indoor farms quickly. While the dim lighting aspect and the light quality of that mechanism is interesting, and although we found it can increase the amount of conductance for lettuce, we did not find that this was at all able to relieve tip burn risk, and so is not a very promising solution to that issue. However, we did confirm what several others have confirmed, that the downward airflow component driving more transpiration from that lettuce crop canopy is highly effective at alleviating tip burn and should be something that we focus on considerably in the future as we continue to try and develop better methodologies for indoor growers to afford tip burn risk in their farms. Okay. So with that, thank you guys very much. I really appreciate you. Time.
Okay,
Thank you.
thank you. We are 5 minutes over the original, but those who want to ask questions, I think we can extend 5 more minutes. Yeah. All right. Questions from this classroom? Yes. Yes.
Maybe green light doesn't have the effect on stomata that blue light does, but supposedly green light penetrates better. Is that why green light wasn't, or why wasn't green light used for the nighttime dim lighting?
Yeah. So mostly I was trying to look at that mechanism, and so the blue and red light mechanisms were pretty well understood, and then green light has had some application, but it's not as effective at driving that specifically blue light phototropin response. It's not as effective at red light at driving the photosynthetic response, and so I wanted to select those two to focus on for that application.
Okay. From what your question?
So we have 2 questions in the
Please.
chat. So Dr. Francis asked, you've collected tip burn data under standard conditions for a number of varieties under replicated conditions. Have you estimated heritability?
I have not estimated heritability. That is something maybe I should look into doing, but I have not done that.
And then there's one from Nadia. She asked, how would you expect dry room conditions, high VPD conditions, to affect your results?
Yeah, so I mean, I would expect that there would be a lessening of that tip burn severity within that environment if there's drier air condition, but once it becomes too dry, the stomates will close and they'll limit the transpiration rate. So I guess it depends on what the point of dry room, high VPD means in that context. Right, but if it's a drier aerial condition, we're going to have more transpiration and mass flow of that calcium, and so we should see a reduction in tip burn severity.
So it seems that industry is doing a better job, like recommending, like, let's say, resistant tip burn cultivars. Do you think this will probably be the most viable strategy moving forward in the next, let's say, 10 years, then like keep like tweaking everything and try to find the best optimum that will probably change between bigger growers, medium, small growers? Or do you think it might be like some sort of combination of the two of them?
I think it'll end up being a combination, right? There's, so we want to develop better germplasm and we want to develop better lettuce cultivars that have lower tip burn risk, but there's also such a wide variety that it's probably going to take time. If the indoor farm industry begins to standardize a bit more in terms of how structures are built and what kind of, if the lighting strategies are more consistent or light module designs are more consistent, greenhouses are often a bit more standardized in terms of how you would build that type of environment. And indoor farms are just often so N of one with how they customize designs. And so if there's less customization, then it's going to be easier to develop and implement strategies that might be helpful, right? But also developing tip burn resistance is something that's also going to be a large factor in resolving this issue. And so it'll probably end up being a combination of the two of those things.
Thank you.
To understand the experiment, I know we asked questions, I think. I don't know.
Is there any, because committee has opportunities on separate time, so any other questions, Jason?
Yes, so JL just asked, do you know the calcium level at which the necrosis appears? Is there a pre-tip burn phenotype physiologically or molecularly that could be used as a warning sign to inform changes in growing conditions?
Yes, that's an interesting question. So from what I found, testing so many different cultivars, the amount of calcium that was in tissues that was tip burn free was totally relative to each cultivar. And there are no relationships between, if the inner leaves have this much calcium between these different cultivars versus the tip burn severity index, there's no relationship between how much calcium is required to prevent the deficiency. It's really N of one for each cultivar. But there have been a lot of studies to try and develop some way to predict that decline in tissue calcium, or like some kind of measurement that we can use to predict when tip burn is going to happen. And the best we've been able to do is predict it like 1 day in advance. And so it seems like once that deficiency occurs, it's pretty immediate within how quickly it shows up in the plant. And so it's very hard to generally identify when that's going to happen and then how much calcium is required to maintain that healthy leaf tissue expansion.
I think there's lots of people wishing you, congratulations. Thank you for your presentation.
Okay, all right, then we're gonna conclude this. Well, thank you so much.
At nighttime, when you turn on the fans, you keep them on during the day. Yeah, it's fine. But
20%.